Hello, AP Biology students, and welcome to my Unit 6 video on gene expression and regulation. What a fun unit. Just like all my other videos, this is not for profit. This is only meant to help students. Images come from OpenStax and Khan Academy. Both of those are excellent options for reviewing and going over this unit. So let's dive in. We're going to start with DNA and go through transcription and translation, mutations, how genes are regulated, gene expression is regulated, and biotechnology. DNA and RNA structure, topic 6.1. This is a bit of a review from Unit 1. Can you describe, students, how the structure of DNA leads to its function? Well, to do that, we really need to think about what is the function of DNA. And that function in the learning objective over here is going to be passing hereditary information from one generation to the next. So let's check it out. Before you can get there, I do think it's nice to review the experiments that led up to the discovery of the structure and function of DNA. There's Frederick Griffith with his transformation experiment with pneumonia. There is Avery, McLeod, and McCarthy. And then there's the Hershey-Chase experiment. I featured a Hershey-Chase experiment here. It's a good opportunity to practice your claim, evidence, reasoning skills. What is their claim? What is their evidence? And then how does scientific reasoning tie it together? Here, you can say that the claim is going to be that DNA is the hereditary material. The evidence is going to be the phages that are labeled. So here, a bacteriophage, a virus that invades bacteria, has its radioactively labeled phosphorus. We know that phosphorus is in DNA and is not in proteins. Here, the radioactively labeled sulfur in this bacteriophage. Sulfur is found in proteins, but not in DNA. What a beautifully designed experiment. Let the viruses infect the bacteria, shake the viruses off, and then spin them in a centrifuge. The cells, and you'll see at the bottom will be the cellular material. And we saw that the cellular material at the bottom had the radioactive phosphorus in it, not the radioactive sulfur. The reasoning would be that the hereditary material that's going to pass on the information to the next generation is that DNA. All right, a couple of things we need to get out of the way before we finish up the structure and function of DNA. Prokaryotes or bacteria have one circular chromosome. They may also have additional plasmids inside of them, whereas eukaryotes that have a nucleus will have linear chromosomes. And this is going to enable multiple parts of origins of replication for when DNA is copied. And in humans, you would have 23 pairs of chromosomes. You would have 23 from mom and 23 from dad in a linear fashion. Here's just an example of plasmids inside of a bacteria. This is called transformation when you take it up. The race for the structure and function of DNA involved many players. Maurice Wilkins and Rosalind Franklin at King's College, James Watson and Francis Crick at Cambridge, and Linus Pauling. And so you saw, if you can read about it, there's quite an interplay here, and there's a lot of instances where scientists are not being good people, right? Like James Watson is a racist. And you can see that her work was taken without her knowledge or her consent. Here's the famous B-51 picture. And I like, there's an article in The Guardian or The Atlantic, one of the two, that's talking about how, not all scientists are good people. But what we have to take away from this is that people can be messy. We need to learn from the science from it, but we also need to have strong ethical standards. And so I think, I'll say this at the end with biotechnology, but this is a ripe time to talk about bioethics and how to act like an ethical person. So let's get into it. And then what James Watson and Francis Crick published, because they were able to see this picture, was about the structure and function of DNA. And they were able to show that there's a sugar phosphate backbone, sugar phosphate. This is a deoxyribose sugar. It's missing an oxygen as compared to ribose. It has a 5' and 3' end in DNA. There's directionality. And these directions are going to be anti-parallel. It's like streets running opposite directions, like on a highway, one direction and the other direction. This lets DNA fit together and make its turns and make its famous double helix. The new strands of DNA can only be added on the 3' end. You'll see on the 3' end, there's this hydroxyl group hanging off, this OH, and that's going to be the area for dehydration synthesis for new nucleotides. There are 4 bases in DNA, and your sequences of bases is what leads to you being you. Now, amazingly, and there's this beautiful TED Talk on it, if you were to print out all the DNA in you and put it inside of books, You know, the only difference between you and your neighbor or your friend would just be half of a book if there were like 20-something books. It's not a lot, but that little bit does give differences between boys and girls and all the different wonderful phenotypes we see. A's and T's will bind together, and so will G's and C's. And you can see it very clearly in this picture. It's going to engage in hydrogen bonding between the electronegative oxygen and nitrogen and the hydrogen on the other side. G's and C's will have 3 hydrogen bonds between them, and A's and T's will have 2. That will come in to play later on during transcription with what's called the Tata box. Purines, I just remember AG stands for silver, and that was always pure in my mind, will be double ringed, and a purine must always match with a pyrimidine. This will give this correct size for DNA. And then you can see here, this is why C's and G's and A's and T's go together, as found out by Chargaff and Chargaff's base pairing rules. The pyrimidines are going to be C's and T's, and then instead of a T in RNA, it will be a U for uracil. So can you do it now? Can you talk about how the structure of DNA leads to its function? The structure being these bases carrying the genetic material. They're going to code for RNA, which is going to code for proteins. The sequence of these bases gives us genes, and those genes code for who we are. DNA can be copied because of the base pairing rules, A's to T's, G's and C's. And it can be read with the directionality with enzymes like RNA polymerase to make transcripts and to make proteins. So what a beautiful molecule that gives us all of our unique characteristics. All right, topic 6.2 is replication. And here it's really important to look at the exclusion statement. They just want you to know DNA polymerase, ligase, RNA polymerase, helicase, and topoisomerase. You can always go beyond that, of course, if you want to. It's really fascinating to read about how DNA copies itself. I would encourage you to spend some time on the Messelson-Stahl experiment, often called the most beautiful experiment in biology, proving that it's semi-conservative. It's another excellent opportunity to engage in claim evidence reasoning. Here it is. And so Messelson and Stahl grew bacteria in radioactively labeled nitrogen-15, right? And this nitrogen-15 was heavier. It was allowed to grow there for multiple generations. And then it was switched over to nitrogen-14. And then they took it out after each generation. You can make this chart. So here's how we figured out that DNA is semi-conservative. So here it is in the nitrogen-15. It was then allowed to replicate once in nitrogen-14. And you can see that it's going to have one strand of nitrogen-15, one strand of nitrogen-14. So in a density sense, it'd be between 14 and 15, 14.5. So that's where all the DNA came out. Now you can start to see why this is called a beautiful experiment. The next round, right, this is going to open up, and this original strand of N15 is going to be here, and a new strand will be synthesized to it. And then a new strand of N14 will be synthesized to this one. And so same here. This N14 will come over here, and we'll have another strand synthesized to it. Because of the base pairing rules, A's to T's and G's to C's, this works, and we get an exact copy each time. There, of course, can be errors, and there's proofreading enzymes for that. And so here you can see after generation 2, you're going to start to have a band that's at N14 level and a band that's at N15. And you should have 50% of them at N14 and 50% of them at 14.5, excuse me. Generation 3, you'll see 75% at N14 and 25% at N15, and so on. What a beautiful experiment to show that DNA replication is semi-conservative. If it wasn't like this, we wouldn't get an exact copy of DNA in every cell. We start as one zygote. And from there, we go through trillions of rounds of cell division in mitosis, and we get exact copies of DNA all throughout our body. There are some permutations to that with our immune cells, with some enzymes called recombinase. But the majority of our cells all have the same DNA. All right, can you model it? Remember, here's the trick. Remember that it only adds to the 3' end. Can you model it given a figure like this? I would encourage you to try AP classroom problems and to try the personal progress check for Unit 6 here. Can you use these enzymes? Here's what it should look like. All right, so this would be the leading strand. So this is just backwards from this one, but that's fine. The enzyme is going to unzip DNA and allow this process to start is called helicase. Topoisomerase is the enzyme above it that's going to help to release the tension from supercoiling of DNA. I've had my students in the past, not this year because of COVID, but take ropes and they help to tie them around each other, and you can see that they get really tight as you start to pull them apart. And so topoisomerase actually relieves that stress. Let's look at this. So this is the 3' end right here. And we are going to use the pointer. And so this would be, it's going to, here you can see the 5'. It goes from 3 to 5. So the other side has to be anti-parallel. It has to be the opposite. So this is the original strand, this green one. Here's the one that's being synthesized. It's going to go 5 to 3. This is called the leading strand. Remember that DNA nucleotides can only be added to the 3' end. So this one works fine. There's an RNA primer put down by a version of RNA polymerase called primase. It's then going to be replaced by DNA with DNA polymerase. DNA polymerase is going to add those new nucleotides. It's going towards the replication fork. This is, everything's going great. It's adding a new strand. It's going to use those base pairing rules, A's to T's, G's to C's. It's going really fast. You should look at some of the animations on HHMI or the DNA Learning Center. Now, a Japanese scientist figured out this lagging strand. So here's the conundrum. This strand is 5 to 3. Okay, so the other side has to be 3 to 5. But here's the problem. It can only be added on the 3' end, students. So you'll be adding away from the replication fork. So this isn't going to work. The way we do it is in short fragments. We still have to add to the 3' end. So it still has to be added to the 3' end of the fragment. So this would be fragment 1, fragment 2, fragment 3. These are called Okazaki fragments after the scientists who figured this out. They're synthesized in short strands and then ligated together with DNA ligase so that they're bonded. So even though it's going away from the replication fork, they're just synthesized towards the 3' end and then bonded together. And then the enzyme picks up again, DNA polymerase, and starts synthesizing the next strand or the next little set. This is called a lagging strand or discontinuous. And so one of the progress check problems really had you draw this out. So could you do it here? Right? And so let's see real quick if I can use my drawing. This is 5', so this would need to be 3'. Here's 3', so this would be 5'. 5' to 3' works fine. 5' to 3', so this is going to be the leading strand. This is the lagging strand, right? It has to go in this direction towards the 3'. This direction towards the 3'. 5. Well, I've now learned that it's harder to draw on this thing than I thought. And so here would be those Okazaki fragments. There'd be ligase to bond them together. All right. Pause for a second. Okay. We are ready to move on. I'm having trouble getting my clicker back. Here we go. And now I need to get rid of these drawings. Pause. Okay, thank you for bearing with me as I learned that on the fly. That was DNA replication. So now we're on to transcription and RNA processing. So we've learned how DNA copies itself. We learned why DNA is the hereditary material. Now we need to learn about the central dogma, right? And from DNA is going to be made RNA, which is then going to make a protein. So when I was hired here at TASM, our school, I was asked to give a quick mini lesson on protein synthesis for someone that's not a biology major. And so I talked about the cookbook. And so how the DNA can be the cookbook, and then you're going to write down the recipe. That could be the RNA. Excuse me. And then you're going to take that recipe and go to the store, and you'll pick up the ingredients, which are the amino acids, to make the meal or to make the protein. So this is going to be the step where we're going from DNA to the RNA transcript, which can then go into the nucleus, or excuse me, out of the nucleus and go make the protein. All right, so can you describe the three types of RNA? Can you model the flow of genetic information from DNA to protein? Can you model transcription with initiation, elongation, and termination? Can you describe modifications that go from pre-mRNA to mature mRNA in eukaryotic cells? And how might these modifications you described in number 4 contribute to protein diversity? Here we go. Here are the three types of RNA. There is messenger RNA, transfer RNA, and ribosomal RNA. It's important to know what each one does. Ribosomal RNA combining with some proteins will help make ribosomes. Ribosomes are ubiquitous. They're in every living thing, and they are the places where protein synthesis occurs. Ribosomes will process mRNA, matching it up with tRNA to get the correct amino acids in order for the protein. tRNA is a type of RNA that has a shape characteristic like a T that will have at its bottom an anticodon, and at its top it will have the correct amino acid. Messenger RNA is what is transcribed from DNA in the nucleus and then goes out into the cytoplasm to be read at the ribosome. Now in bacteria, there is no nucleus, right? So it can be transcribed and translated at the same time. So their names kind of give them away. Messenger RNA, it's carrying the message of the DNA. Transfer RNA is transferring the amino acids. Ribosomal RNA, it's making the ribosome. Prokaryotes do not do anything special to their messenger RNA after they make it. There is some type of splicing, et cetera, with transfer RNA and ribosomal RNA. But let's go on. Here's that central dogma. So the DNA is here, and then the DNA is going to be transcribed into RNA or messenger RNA. That messenger RNA at the ribosome will be translated into a protein, and the protein is going to be dependent upon the sequence of code in the RNA. Here is kind of a more in-depth picture of it and showing some directionality. You can see here that transcription, just like synthesizing a new strand of DNA during DNA replication, is only added to the 3' end of the RNA transcript. That means that the DNA is read from the 3' to 5' end by this enzyme RNA polymerase. In eukaryotes, not in prokaryotes, but in eukaryotes, there can be some processing where introns are removed from the pre-mRNA into the mature mRNA. And that's something I needed to emphasize more with my students. The mature mRNA can then be sent into the cytoplasm to be made into a protein. In the ribosome, there will be areas where 2 tRNAs can fit at once, and their anticodon will match up to the codon in mRNA. All this costs energy to do translation as well. Here's some more vocab for you. And so this is going over the coding strand and the template strand. The template strand is called the non-coding minus strand or the antisense strand, depending on what you're dealing with. And it's the strand that is being read by RNA polymerase to make RNA. The coding strand matches the RNA. Look at that. ATG, ATC, TCG. Well, it matches. The only difference is that there's going to be a U instead of a T. And so the transcript will ultimately resemble the coding strand. The template strand is the one being read by RNA polymerase. Here would be an example of it. RNA polymerase will be reading this 3' to 5' template strand and making its 5' to 3' transcript here. There's 3 steps. There's initiation, where it's going to bind on with the help of transcription factors. This takes place at the TATA box. TATA is important because, look, there's only 2 hydrogen bonds between thymine and adenine, as opposed to 3 between guanine and cytosine. So it's easier to open up the DNA here for transcription. It will elongate or make the transcript, and then will terminate once it reaches the end of the sequence. Okay, so now going from that pre-mRNA, so this was made by RNA polymerase, to mature mRNA in eukaryotes. Notice how in prokaryotes or in bacteria, it can be transcribed and translated at the same time. Here, you can see this in this nice picture here. RNA polymerase read that DNA gene, it made the mRNA, but it's going to be processed before it goes off for translation. So this would be pre-mRNA, and once it's done being processed, it would be mature mRNA. There's going to be a 5' cap added. It's a guanine cap to the 5' end. This is going to help it attach to the ribosome. It's going to have a poly A tail, which helps to protect it in cytoplasm from enzymes that are chopping it up. The introns will be removed and the exons will be left there. This, I remember it because exons are expressed and introns are not. And there can be what's called alternative splicing. And so here we have 30,000 genes, but we have many more proteins. And so sometimes you could have, in this mRNA, you're going to have 1, 2, 3, 4, 5 exons. Here, they're going to leave out exon number 3. And here, they're going to leave out exon number 4. And that gives you different protein products. There's a really nice question about this. You should ask your teachers to look it up in AP Classroom on the FGF5 protein. All right, translation. So we've read the DNA and turned it into an mRNA transcript, a mature mRNA transcript. Now, it's going to leave the nucleus, and it's going to be translated at a ribosome, or in a prokaryote, it's translated at the same time. So what's going on? First of all, you need to remember a bit from Unit 2 about the endomembrane system, about ribosomes. So we're going to review that. You need to know their location. You should be able to describe the phases of it, but you don't need to know all the enzymes in it. You need to describe the special case of retroviruses with this central dogma. So here's the endomembrane system, if you remember from cell structure and function. These ribosomes on top of the ER will receive the mRNA transcript from the nucleus. They'll synthesize proteins, and then they'll be sent to the Golgi apparatus, where they'll be modified and then sent out of the cell. If they're free-floating ribosomes, then those proteins will be made as well, but they'll be used within the cell. And here would be an example of free-floating ribosomes. These are in eukaryotes. Notice how bacteria have ribosomes as well, right? It's not a membrane-bound organelle. So bacteria have these ribosomes. Ribosomes are in all units of life, and they will also synthesize proteins at them. One thing I think you can see here, right? You can have multiple ribosomes on an mRNA transcript. So you can make multiple proteins. All right, initiation, elongation, and termination. This is a good time to review the 3 types of RNA. There's ribosomal RNA making up the ribosome. There's a small subunit and a large subunit. mRNA was what was made in transcription from the nucleus or from the DNA. tRNA is carrying the amino acids. This takes energy for this to happen. All right, and so here the codon is going to be read in sets of 3 letters, 3 nitrogenous bases, and match up with the anticodon. And it uses the base pairing rules. A's to U's, A's to U's, G's to C's. All right, and then the tRNA will carry the correct amino acid there. It will exit through the E site, and there can be 2 tRNAs in here at a time. So initiation would be when it starts, elongation is making the protein, and termination is once it reaches the stop sequence, like a UAG, for instance, and it cleaves the protein and lets it leave the ribosome. Same terminology as transcription. Here is just an in-depth picture of tRNA and rRNA and their shape. You need to be able to read the genetic code, the table. You do not need to memorize it, right? They will give this to you on the exam. Many of you might have learned in introductory biology the circle, but in AP biology, they're going to most likely give you the table. So the first letter would be here. Let's look up AUG. It'd be in this row. U would be in this column. And then you look for the third letter, G. So AUG codes for methionine. Let's look up CAA. CAA-GLN. All right. And so you can look them up. You can see tyrosine, cysteine. You can look up a bunch of them. But what you can start to see is that many of them will have the same first 2 letters, but the third letter may be different. This is called the wobble hypothesis, and it shows that the genetic code is degenerate or redundant. If you look at this, there's 20 amino acids, but yet there are 64 combinations of the 3 letters in the codon and anticodon, right? And to get that, you do there's 4 letters, A's, U's, G's, and C's, and you do 4 to the 3rd power. So 4 squared is 16 times 4 is 64. So 64 combinations, but only 20 amino acids. So some of them have to code for the same thing. But notice how the first two letters are always really important. The third letter can differ. And this is probably to lessen the impact of mutations. And so here is just an example of it from Khan Academy of the wobble hypothesis. So here, AAG, UUC, that can still work. It can still work here for phenylalanine, but it wouldn't work for leucine. All right, so what was important there? We'll come back to that in mutations. A retrovirus, an example of this would be HIV. HIV infects cells and it's going to actually integrate its genome into the cell's genome. But it's an RNA-based virus. So it has to go from RNA to DNA. And so we have learned how to use this enzyme to take RNA transcripts and make the cDNA or the complementary DNA to it. So here would be the viral RNA coming into the cell. It's going to use reverse transcriptase to make a DNA copy. There will be another enzyme called integrase, which is going to help to integrate it into the host DNA, and then it's in there. Once it's in the host DNA, it can express this gene to make new RNA for new viruses and to make new viral proteins. And it's really hard to fight. And so here you can see an example of reverse transcriptase. And this would be the opposite of the central dogma, right? Central dogma went DNA, RNA, protein, but here we're going from RNA to DNA and then DNA, RNA, protein. Pretty cool. All right, regulation of gene expression. So if the vast majority of our cells have the same DNA, then how do we have different cell types? Can you compare and contrast regulation of gene expression in prokaryotes and eukaryotes? I think that this is a fascinating topic. And there's some really good items on HHMI with stickleback fish that you should check out for this one. So let's look at this and think about what steps can these processes be regulated. For prokaryotes, it's going to be primarily at the transcriptional level. So taking DNA and coding it into mRNA will be the main way that this can be regulated. And we'll see that with operons. Eukaryotes can regulate at many levels. So we can regulate at making the transcript. We can regulate from going from pre-mRNA to mature mRNA. We can regulate once it leaves and before it's translated at the ribosome. We can even regulate the protein once it's made. Most regulation happens at a transcriptional level because that's going to conserve the most energy. Remember, it takes energy to do these processes. So if we need a lot of gene 3, maybe it's transcribed the most, and maybe we don't need any of gene 4. Okay, here is the lac operon. This is called an inducible system, or one that can be turned on. So can you model what's going on here? Take a second, pause the video, see if you can figure out what's going on. All right, I hope you're able to model. Up here is going to be a section of DNA that's going to code for a repressor protein. It's going to be transcribed and translated, and it's going to make a protein that's going to attach to DNA here and repress the transcription of these genes. This repressor will be removed if lactose comes into the system. So if lactose comes into the system and then the cell needs these genes to be expressed to make the enzymes to break down lactose. So it needs to be able to eat the lactose. So you eat your sugar, your ice cream, or your milk. It comes in, the lactose comes and it binds the repressor. It's going to remove the repressor. Watch those arrows. Once the repressor is removed, the RNA polymerase can transcribe these genes. These genes, beta-galactosidase, permease, and transcetolase, are going to help to digest lactose. So how cool is that? This is like you walk in a room and you turn on the lights because you need the light. Right? If there is no lactose there, then the repressor is present. And these genes aren't transcribed, which is going to save the bacterium energy. Let's add another level of complexity to the model. Here is a protein called CAP, catabolite activator protein, and it's going to be activated by CAM, which is going to be activated when there's really low levels of glucose in the cell. Glucose is a monosaccharide, whereas lactose is a disaccharide. So glucose is already broken down one level, so it's easier for the cell to use. So the cell would rather process glucose instead of lactose. So if glucose is present, then CAP is not activated. And it would say that there is lactose present. Well, that lactose will take away the repressor, so transcription can happen, but it'll happen at a low rate, because what CAP does when it is activated is it helps to promote transcription with RNA polymerase. It helps to make the process go off better. So it helps to achieve higher levels of transcription. So CAP enables the process to happen. So here, if there is no glucose, then there's going to be higher levels of cyclic AMP in the cell because ATP has been used up. That CAM is going to bind with CAP, which will bind at the CAP site and promote transcription of the lactose genes. So glucose absent, lactose present. Now, if glucose is absent and lactose is absent as well, you're kind of out of luck, right? You need an energy source. But here there is an energy source, it's lactose. The preferred available energy source, glucose, is gone. So therefore, the transcription is going to be amped up. You can see by the shape of the arrow from OpenStax. So lactose is there, it removes the repressor, and it's going to happen at a higher rate because it's being enabled by CAP, because CAP was enabled because of glucose. So just showing another level of complexity to the model. So can you build your own, right? And you can use this chart to help you practice it and go through it. All right, that was an inducible system, or one that we can turn on. Let's see one that's always on and that we turn off sometimes. So here in Oman, it's usually very hot. And so we run our ACs a lot. But if we get a cloud or a cold spell for some reason, then it's a nice instance where we can turn off our air conditioners. Well, here, the TRP operon, we need to constantly make tryptophan. And so these sets of genes are going to code for enzymes to help in tryptophan biosynthesis. So this will always be on. But if the cell eats something that has excess tryptophan in it, then we can block transcription of these genes, right? Save energy. So the cell ate something with tryptophan in it. Tryptophan will activate the repressor and stop transcription. Let's say that there isn't tryptophan, then things will proceed as normal, right? If there's lower levels of tryptophan. But if the cell somehow came into some tryptophan, then it can activate a repressor and turn this off for a while. It's like if it got cool enough, we can turn off the AC for a while here in Oman. Those are prokaryotes. You should model those. There's a really good POGO on it if you use POGOs. Let's talk about eukaryotes. Eukaryotes don't have operons like that, but we have a lot of ways that we can control it. The first can be the actual access to DNA. We can tighten it up with methylation, adding a CH3 group, which is going to make it so that it is unable to be transcribed. Or we can loosen up the DNA with an acetyl group, and this histone acetylation is going to make it more loose and able to be transcribed. We can then regulate at transcription. And once again, this is the major level of regulation. And for example, here, this we did a model of this with the pituitary switch with stickleback fish and whether or not they had those pelvic spines. So if there's transcription factors that can activate the transcription, then it'll go on. Like the DNA will actually hairpin, bend, and come back on itself. But if there are no factors, then it can't proceed. For instance, in the eye, let's say they were transcribing a gene that's important to the heart, to heart cells. Well, that gene might not be important in the eye, and we don't want to transcribe that gene and make that protein. So in the eye, there would be no activators. There would be no purple, blue, and red one to make the DNA bend like this and come back together and transcribe the gene. So even though the eye and the heart have the same DNA, the transcription factors, which are proteins themselves, are only present in the heart for the genes that are specific to the heart. This is how you can get differential gene expression. And this is really important. You can also have activators and repressors. So an activator could come in and help get this transcription process started. Or you can have a repressor saying, no, we do not want to transcribe this gene. We've already gone over RNA splicing, but this is another level of control that could happen to make different protein products. There's a large sequence of the genome that's non-protein coding, and we don't quite know all of what it does, but we think a lot of it codes for microRNAs. And microRNAs like slicer and dicer can inhibit translation. So even if mRNA is made, but we don't want it to be turned into a protein, we can use microRNAs to actually slice them up and dice them up and prevent translation. So this type of regulation would let transcription happen or not. A microRNA, the transcription's already happened, but we can chop up the RNA before it gets out into the cytoplasm to be read at a ribosome. There can also be translational regulation at the ribosome. So EIF2 is a protein that helps the ribosome assemble on the mRNA. And when it's phosphorylated, translation is blocked. The ribosome can't assemble on the mRNA transcript. When it's not phosphorylated, translation occurs. So this is another level of regulation. And also post-translational regulation. So you can actually phosphorylate, and which can activate or inactivate proteins. Thinking back to Unit 4 with cell communication and signal transduction pathways. We can also mark proteins for destruction and for recycling with ubiquitin. And so if you mark it with ubiquitin, then it's going to go to the proteasome, which is like a recycling center, to chop up these proteins so that the amino acids can be used again. So think about all the levels of regulation that we had there. You had whether or not the DNA can be accessed. You had pre-mRNA to mature RNA splicing. You also there had the 5' cap and the poly A tail. And there's some research that shows like the longer the poly A tail, that the longer the transcript might stick around. You then have slicer and dicer, these microRNAs that can prevent the mRNA from being read at the ribosome. You have proteins that can actually influence whether or not the ribosomes can bond on the mRNA transcript. And then last but not least, even if the protein's made, you can mark it for destruction and for recycling, or you can phosphorylate it to activate it even further. So you have lots of levels of regulation inside the cell in a eukaryote. All right, mutations. So can you model the following? Frame shift, insertion, deletion, silent mutations, missense, nonsense mutations. Can you use natural selection to describe why mutation is random, but natural selection is not? And lastly, can you describe transformation, transduction, and conjugation in prokaryotes? Let's dive in. Here is what many teachers use to teach mutations. The fat cat ate the rat. Notice how they're all 3-letter words, right? So they mimic codons and anticodons. So here, if we insert a letter, I'm going to insert this B right here. It changes the whole sentence, right? And it's called an insertion. And it's going to change all the amino acids. You can see this example here after. And so that could be very deleterious or harmful to the protein. It could be useful to the protein. Here, I'm going to delete the A in fat, right? And so then it changes the whole sentence as well. This is called an indel, and insertion or deletion is called a frame shift mutation. Here, we're going to have the original DNA is going to be TTC, and we're going to mutate that third base to make it TTT. So we go from AAG in the mRNA to AAA in the mRNA. Well, due to the wobble hypothesis and due to the degeneracy of the genetic code, both of these will code for lysine. This is called a silent mutation because the amino acid hasn't changed. It will have no effect on the protein, but this could be passed down, right? It could be, if this happens in a cell, a germline cell that's going to give rise to a gamete, a sperm or an egg, that it can be passed down to the next generation. A nonsense mutation, well, what does your mom tell you if you're engaging in nonsense? She probably tells you to stop. A nonsense mutation is when the DNA, TTC, is changed to ATC, which has changed the mRNA into a stop codon, UAG. It's going to cleave or cut off the protein early. This is usually harmful. A missense, it's just like it sounds like a mistake. So here, the TTC becomes TCC. It becomes, instead of AAG, it becomes AGG. And this is going to code for arginine. Now, this is a conservative missense mutation because arginine is close in structure to lysine. It might work the same. You can see it's a little different, but they're both basic R groups. Here, non-conservative is going to change it a lot, right? And so it went from TTC to TGC. It becomes ACG, which becomes threonine, which is going to be a polar R group as compared to these basic ones. So it's going to be different. And so this is an example of this would be sickle cell anemia with valine and glutamic acid. Now, some of these mutations could be more helpful. And so there's been evidence of that with some mutations with HIV, protecting cells against HIV. Now, the key to this is that mutation is random, but natural selection is not. And so natural selection will sort out the winners and losers of the mutation game. Okay, let's look at genetic variation in prokaryotes. So plasmids can be taken up through the process of transformation. Many teachers do the bacterial transformation lab with this unit, and it's a great time to learn about plasmids and transformation. This would be a horizontal acquisition of information, right? We as humans pass down our genetic information vertically from generation to generation. Here they can pass them along to friends, and so bacteria can evolve very fast. Bacteria are constantly in battle with bacteriophages, viruses that infect them. And so here you can see what's called transduction. And that's when a bacteriophage will bring DNA from a different bacterium and insert it into another one. And that can be integrated into the host DNA. So here's some of this blue DNA. It got inserted into this cell, which can then insert it into its purple DNA and can pass it on to the next generation. So another example of horizontal acquisition of information. Conjugation is when they can pass along plasmids between each other. And then there's transposable elements. And this is kind of called jumping genes. And this is in eukaryotes as well. And Barbara McClintock figured this out. And so genes can sometimes be moved around. And so this was an example of one where it's moved from the bacterial chromosome into a plasmid, which then can go off to other bacteria. I think the College Board will give you context clues on this, and you will need to then say what happens with regards to natural selection. All right, we're kind of running out of steam here, so let's finish up strong with biotechnology. So this is a module that I got to experience as they were putting it together at the University of Pennsylvania. And they invited Philadelphian teachers to come to it. It was absolutely fascinating. I loved it. And I got to go to lots of these genomics talks. And it's fun. I can look at videos and actually see myself in them. That was when I had short hair. And I weighed more. So it's fun to see the change. But they go over how do scientists work with the genome. And so this is interesting, right? They have this exclusion statement here. The details of these processes are beyond the scope of the course. The focus should be on the conceptual understanding of the application of these techniques. So for my students, I really had them, read about it, and then we really looked at a bunch of sample problems, figuring out, well, how can we use these? And so let's start with PCR, right? And PCR is in the news because you're thinking about PCR COVID tests. And so the way it works is you heat up the DNA to separate it. You then cool it down so that the primers can bind to the broken apart strands. And so the primer would be specific to what you want to amplify. So for example, if we're doing a COVID test, it would amplify viral genes, not your normal genes. And so if your sample that was taken from your nose or your throat had the viral genes in it, then it would be amplified. And if not, then it wouldn't be amplified. So you insert these primers and then you heat it up again to the level where Taq polymerase, which is a DNA polymerase, can synthesize new strands. And then you can repeat this cycle 25, 35 times to get lots of copies of the DNA. And I was really excited. I ordered my mini PCR this year and it didn't come. Boo. All right, so here we go. You can replicate it many times. And then you can load them into electrophoresis. And so this is going to be agarose gel. And this agarose gel has tiny holes in it that will allow the DNA segments to move towards the positive anode. DNA is negatively charged, so it will move towards the positive end. Once you turn on the power and it passes through it, you can see the smaller transcripts will be, the smaller segments of DNA will make it further through the gel once you turn it off. The smaller ones will go through it faster. So here's the result of a PCR, and let's say that the primers selected for a 400 base pair length segment of DNA. Once we put it through the gel electrophoresis, we have a known DNA ladder over here, and we pause it. It will come out here right along the 400 base pair. So let's say that we have different markers. And so this is where I think it's worth it to do some problems. And so here we'll see that here's DNA from a crime scene, and it's going to all come out at the 200 base pair length. So this person, because they're diploid, they have a chromosome from mom and dad, are going to be homozygous at the 200 base pair length. They're going to have both number 1 alleles. This person, suspect number 2, will have a 200 allele and a 300 base pair allele. Remember that the smaller ones go through further. And so they're the primer hit for the 300 base pair and for the 200 base pair. So they are heterozygotes. They might have gotten one version of the allele that matches the DNA from the crime scene, but they have a different one. This person is homozygous for the 300 base pair allele. This person is homozygous for the 200 base pair allele. So if you do this in enough areas of the genome where there are variations, then you can get with pretty good certainty that the DNA matches. And so there's a good innocence project that's linked to that's helping to get people out of prison that didn't commit the crime, and that we can see through DNA evidence. Right? Or conversely, you can use DNA evidence to prove someone committed the crime. Here is the transformation experiment. And so this is a great time for you to practice positive and negative controls with regards to this experiment. You'll try and heat shock and get the plasmid in, and then you can grow it in an antibiotic environment. And so what that will happen is if it picks up the plasmid, then it will grow on the antibiotic plate. And then you can grow it on another plate that might help to drive the target gene. So maybe it's arabinose, and that arabinose will make it have red fluorescence, and it'll glow red. And so it's a cool experiment, and it's very helpful because if we can do this, we could grow things like human insulin or human growth hormone via bacteria instead of taking them out of animals. And students, that's a wrap. Have a good one and crush Unit 6. See you for Unit 7.